Nucleus. The human nucleus is filled with chromatin, well, with DNA, and so it's like a highly packed and crowded environment. And of course, even before condensate exists, it is known there are lots of nuclear bodies called nuclear domains, and that are filled in the nucleus. And some of them are on the chromatin, some of them are between the chromatin. So there's a lot of, and they're involved in a lot of chromatin functions. So a lot of work has been done trying to understand the interplay between the chromatin and those nuclear bodies. And of course, the concept of phase separation allows us to ask more questions or use a fresh perspective to look at those relationships. And so today I want to share with you some experiment data that we have in an attempt to address some of those questions. The first one is, how does the chromatin mechanics affect condensate growth? Because the chromatin is a viscoelastic network, and so they're proposed and tested already with experiment to suppress different kind of growth modes of condensates. So how do condensates navigate that? And the other one is, for the condensates that form on the chromatin, what are the cues that allow them to be formed specifically at the right place, at the right time? How do cells regulate this process? And of course, how do condensates help chromatin functions? So the first question is asked by my graduate student, François, and the way we approach this question was to form synthetic condensates in the cell, in the nucleus, and then track their growth over time. So as shown here by this movie, where we form condensates with different properties that we have measured to have different properties, and then we track their growth, and you'll see that has shown the movie and the quantifications. And the one on the right just keeps growing, and we call it continuous growth. And then on the right one, you will see lots of osteoripening, where the small condensates disappear and because of osteoripening. And to make sense of this, we collaborate with our colleagues in physics to define on our regimes that you will have stable growth, and under what we will have ripening in this case. And although theory predicts elastic ripening, I want to say that we've never observed elastic ripening for all the condensates that we look at so far. So it's a suppression of osteoripening most of the case. But that depends on the condensate property. What does it mean for the cell? So we think that it illustrates that the protein condensates, just by the protein interactions, can generate surface tensions in the range that they can counterbalance the chromatin and stiffness, so that cells can actually regulate the condensate growth by controlling not only the chromatin heterogeneity or chromatin stiffness, but also by just making different condensates to control the growth rate, or how they grow, or even where they nucleate. And so for the second and third question, I want to show you the work that we did with PML bodies. And so PML bodies are those condensates float around, actually shoot around. If you remember Danny's movies, they shoot around in the nucleus because they're mostly not attached to any chromatin. They're just freely moving around. But sometimes they do. They're associated with chromatins, but only occasionally and dynamically. And when they're associated with chromatins and they find to have a lot of components, even some of them that are not associated with chromatin, they have some chromatin-associated proteins in them. We don't know why and how they're associated with chromatin, and what do they actually do, particularly the formation of the body itself is functional or not for the chromatin function. And so there is a very stable association with chromatin, and that is what we're going to study. And so the stable association is the localization of PML bodies to cancer cells on telomeres with some specific cancer cells. And this is so stable that if you just stain for the cell or cancer tissue, you will see this co-localization between telomere and the PML bodies. It is so unique of those cancer cells that they're actually used as a diagnostic marker in the clinic. And so, so what is all cancers? Why? What's special about them? So ALT stands for alternative lengthening of telomere pathway. And so telomere lengthening pathway is required for cancer cells to keep dividing, because normal cells divide and then the telomere will shorten as cell divide. And so for cancer cells to keep dividing, they have to activate telomere maintenance mechanism to add the shortened telomere back. So, and therefore it is of interest to target this telomere maintenance mechanism for cancer cells because it's so universal to all kinds of cancer cells. And majority of cancer cells just reactive telomerase, which is active in our stem cells, but suppressed in somatic cells. They simply reactivate this gene, and then they get this telomerase, which uses its own RNA, it has an RNA in the complex, as a template, and to do reverse transcription, add the telomere DNA back. But the subset of cancer cells cannot do this. They could not activate telomerase. They use this alternative, just means lack of telomerase. The name itself implies. But what's known about this is that it uses a DNA repair mechanism. It uses telomere DNA as a template to do homology direct telomere DNA synthesis instead of using RNA as a template. So this is a DNA repair mechanism, which is unique because the DNA repair is normally suppressed on the telomere ends because the cells want to prevent the linear telomere DNA being recognized as a DNA break. So this is a particular mechanism that is activated in those cancer cells. And so the unique marker of those is in the localization of PMO body to those cancer cells. It is so unique, it has its own name. It's called alt telomere associated PMO bodies, so APBs. And so what's unique about them is first they have telomere clusters, right? Not only they are localized to telomeres, they have multiple telomeres in them. And so this telomere clustering is assumed to because the DNA repair happens in these APBs, in these PMO bodies. So the clustering is to help provide repair template. And you have a higher enrichment of DNA repair factors in those PMO bodies compared to normal PMO bodies, just more. And also you have this RNA, which is transcribed from telomere repeat and TERA in those condensates. And normal PMO bodies has no detectable RNA content in them. So this is a unique properties of those PMO bodies. And so we want to know how they gain those particular properties and are any of those useful for the cancer cells. And so, of course, our model is phase separation because previously it has been shown that DNA damage on the telomeres then can mimic this alt pathway in the alt cancer cells and can induce APP formation. A lot of the pathway can be induced by artificially inducing DNA damage on the telomere. And simulation is required for APP formation. That's already known by doing a screening method. And of course, PML contains sumo enzyme, and sumo enzyme leads to phase separation. So the hypothesis was very simple, is upon DNA damage, just like any regular DNA damage, you will have stimulation of DNA repair factors on the telomere. And then for some reason, those telomeres now can, because of sumo, it will recruit PML proteins uniquely only here, but not in other normal DNA damaged foci because they don't have APBs or PML bodies. And somehow they can recruit PML protein because PML contains sumosin through sumosin interaction. And now you have enough local high concentration of PML, naturally you get condensation, and that leads to the formation of PML body, and then you form APBs. And because the APBs now, you have a blob, you can fuse, and that will allow telomeres to cluster within them. And of course, the condensation process helps enrich those DNA repair factors that would normally be diffusive, just like a DNA damage foci. But again, we don't know why they use PML in this case. And that will lead to homology-directed DNA repair and thus telomere elongation in them. So it's a simple model, but how to actually demonstrate each step is not straightforward because the cell is very complex. For example, if you introduce DNA damage, you get everything. So how do you know it's the condensation in that cluster? So we use a bottom-up approach to do this. So what we do is we artificially enrich sumo on the telomere without DNA damage and to see what can we gain by simply having sumo on them. And so this we collaborate with David Chenoweth and develop this chemical dimerization system where you have a small molecule that will simply dimerize two proteins together. And so if we do this by tethering sumo or sim to the telomeres, we can produce movies like this where you can see the sumo is enriched on the telomere first, and then you will start to see signature of phase separation there around, and they start to fuse together. And because they are tethered on the telomere, that leads to telomere clustering that we could quantify and could verify with telomere DNA fish, for example. And This depends on the sumo-sim interaction, because if we use mutant of sumo-sim, we cannot induce phase separation or telomere clustering. And interestingly, the telomere clustering can be induced by tethering other phase separation proteins, including different IDRs, but APP formation can only be induced by tethering SUMO. So there's this intrinsic link between SUMO and the APP, but the clustering only leaves some condensate forming on the telomeres. So we conclude that that the simulation likely leads to the phase separation from APPs and the cluster telomeres like we hypothesized. However, the APP detection was done with immunofluorescence, and we did actually not follow PML. So we don't know actually whether the simulated telomere actually recruit PML. And so the reason that we need to verify that is because in the field, people believe is that telomeres gets recruited to existing PML bodies, other than what we propose, that if the telomere actually nucleate de novo PML bodies to form APPs. So to do that, then we need to follow APP formation kinetics with live imaging, but we have to do this in endogenous PML level because of phase separation is so sensitive to overexpression of proteins. So we CRISP tag PML, and then my student Rongwei did this live imaging, where we tether again SUMO just to induce artificial PML body formation on the telomere, and then she produced movies like this, and we follow here the telomere is not labeled because we use the color to label PML, but the SUMO is always localized with telomere, so we use it to replace telomere signaling. And so she quantified, just sit in front of the computer, stare at these movies, and then she can see those kind of events. And the first one we call APP fusion, because she can see a SUMO dot, see a PML dot, and then come merge. And then she can also see where sometimes you see a SUMO dot, and the PML just gradually increase intensity. You don't see another PML body fly over and then merge with the existing SUMO dot. And we call that a nucleation model. And so to confirm this is not just the artificial SUMO system, we used what's commonly used in the field to induce odd phenotype is to tether a nucleus FOC1 to the telomere, and you do this fusion protein, and it will cause damage on the telomere, and then you can cause APP formation and all other odd features that have been already studied in the field. And so again, we do the same experiment, follow the movies, and we get similar effect where you see both fusion and the nucleation. But if you quantify them, first of all, the quantity is different. And if we look at SUMO versus FOC1, which is the first group here, FOC1 is much, much less in the endogenous system. And so, but when we do the endogenous, we get a lot of APP formation in general. And also the nucleation, which is the blue one, is more than the fusion in the SUMO recruitment case, but not in the endogenous system. So meaning that in our artificial system, by artificially boosting the amount of SUMO there, we are amplifying the nucleation. So that indicates nucleation correlates with the level of SUMO on the telomeres, but not fusion, because if we use mutant, we can still get similar fusion. And we can do this with SIM, and also the isoform, which SUMO we use doesn't matter, meaning that it's this level. You just need enough SUMO to attract, maybe compete with the existing PML body for free PML to nucleate. It doesn't matter what SUMO, how you get there. You just need SUMO to be there. So, and consistent with this, if we look at the telomeres that form APPs by fusion, and we'll see that the ones formed by nucleation always are bigger and brighter. So that correlates with how much SUMO, because both of the systems use tethering and depends on the TF1 level on it. So more telomeres, you just get more fusion. So meaning that, again, correlates with the SUMO level. And also, if we look at the time it takes for either fusion or nucleation, we consistently see nucleation happens faster than fusion, even though there's a lot of variance in the data. And if we try to do this on the centromere, and interestingly, if we do DNA damage, we don't see APP from PML body. This is not called APP anymore. It's just de novo APP PML body on the centromere. But we can see this when we dimerize SUMO. So, and has quantified in here. And this is because that if we do damage on the centromere, it actually cannot enrich enough SUMO that's detectable, right? Even though we believe SUMO nation should still happen like any DNA damage, but it's just not enough to actually have enough sumo on the telomere to form PML bodies. So that means that we have to redraw the model where not only that PML can be nucleated on the telomere, it can also be fused with a sumo-enriched probably condensate. If it's a sumo condensate, then we wonder what are the proteins that can form and can be in the condensate that the sumo telomere can attract and allow them to fuse with the PML body. And of course, naturally, we think about DNA repair factors because they're known to contain stimulation sites, particularly those ones involved in homology-directed repair, and they don't depend on any PML function as far as we know. So we tested those, but we do this in the PML knockout cells in order to decouple the contribution from the PML pathway. And so in those cells, then we don't have PML body, and then we don't have APBs, and then we just do the same thing, we tether, and then we can produce this kind of clustering events, and but you can see the degree is less than the white type cells, right? So PML still is very important, even for this kind of artificial system. And we can see the enrichment of DNA repair factors without PML, and we look at like helicase, like balloon, RPA, RAD52, AP1, lots of them can be enriched. And then that means that, is the DNA repair factors that can be enriched on the stimulated telomeres to cause the telomeres to be sticky to PML bodies and allow them to fuse with PML bodies. And then we tested that. Now we asked this, does the formation of APB actually promote DNA synthesis and telomere elongation like we hypothesized? So to do that, we use the EDU chaser, which is a homologous thymidine that can be fluorescently labeled to look at newly synthesized DNA, and then we look at non-S phase cells, so we're not looking at replication, and then we look at localization with the telomere signal, where we visualize with telomere DNA fish. And so this is a PML knockout cell recruiting sumo. You see some of those kind of dots that localize telomere, and you don't see when you do the different variants of the sumo mutant. And then the quantification shows here. So in the white type sumo tethering, you induce a lot of newly synthesized telomere DNA, and the PML knockout cells, sumo tethering can still do it, but again, is much less than the PML containing white type cells. So meaning that PML is very important, still important, even though it's not required. And then the mutants are important. So the sumo-semi interaction and the ability of sumo to be conjugated to protein is important, meaning it's those DNA repair factors that are important for this process. And then what about endogenous cells, right? Those are all artificial. So we synchronize cells, the alt cancer cells in the G2 phase where the telomere synthesis is active. And then we're trying to apply sumo inhibitor, that's an E1 inhibitor of stimulation, to cells with PML body or to cells without PML body. And then we look at the EDU synthesis, and the quantity is low, but we can quantify them nevertheless. And we can see in both cases, the sumo inhibitor will reduce the amount of telomere DNA synthesis on the telomeres. But again, if you just look at the white type compared to the PML knockout, endogenously, you still see more in the white type than the PML knockouts. That leads us to conclude that this telomere elongation-based pathway is the sumo-mediated DNA repair factors that is doing the job, but the ability of telomeres that to access PML body really helps boost this positive feedback loop, because we also have data show that if you lock down sumo, you actually reduce the amount of sumo on the telomere. So it's like a positive feedback loop that has been set up by the PML body that being evolutionally selected because they can gain access to PML body. So consistent with probably all the condensate function that is not required, but it really helps. So kind of boring conclusion, but here we are. Then for RNA, what do we do about the RNA, right? It's puzzling that it doesn't have RNA. And then our intuition is it comes with the telomere, it doesn't come with the PML body. PML body happens to be RNA, but it might play some role of forming the PML body. That was the hypothesis. So to test that, we hypothesized there must be some RNA binding factors, proteins that are important. So we knocked down Tera with ASO, and then we use this DNA damage assay to screen for proteins that needs Tera for localization. And by doing images like that, we have control, and then we have Tera locking down, and here showing the LSD1 protein. And you can see that the localized to telomere is fine upon DNA damage, but if we knock down Tera, it's gone. So it really depends on Tera for its localization. And we also find EZH2, but LSD was really most dramatic. We don't find like non-low-fast. It's just not very obvious in the system. And anyway, so and then because Tera and LSD1 interact, and of course you have to see whether they form a blob. So we did that, and with our collaborator who is expert on LSD1. And this blob formation depends on Tera G-quadruplex conformation. And interestingly, if we use a G-quadruplex inhibitor, it doesn't abolish the condensate formation. And we show that this interaction promotes R-loop formation, and it's important for APB formation and other features, so meaning that it agrees with the hypothesis comes upstream of the APB formation, but we don't know how it contributes to APB formation. So, to summarize, I've showed you that our synthetic condensates tracking data show that the condensate property can play a role in determining how condensate grow in the nucleus. And by using PML body example, I show you that RNA and post-translational modifications can be used to nucleate condensates on the telomere, and those condensates help concentrate repair factors and then bring telomeres together to help the DNA repair. And of course, lots of open questions. And for example, we're interested to ask how do chromatin associate condensates cluster with each other and or with the existing nuclear bodies? Is through passive process, or actually cells regulate this process for some of the applications, or how do RNA and the post-translational modification work together to regulate the condensate nucleation or growth or composition? And then, in particular, of those R telomere, we're interested to ask what are unique features about those R telomere that allow them to reach this critical simulation sumo concentration for nuclear PML body that other DNA damage sites that don't have. And of course, so far, I show you data of more qualitative description, but which is not very satisfying. And answer those questions, we want to be a little more quantitative. And of course, these bridging between experiments and theory. But sadly, despite having hundreds of bridges in Pittsburgh, those bridges are not sufficient. So I'm very grateful for this program and the conference to make it possible. So thank you.
That was very cool. So I'm just wondering, so is it possible then that simulation in the nucleus generally is, a signal for clustering things? I mean, like, there's many, like, for example, chromatin, histones are simulated, lots of things are simulated. And is that, like, is PML a way to bring these things together in one place? I mean, also when you have homologous recombination, you need to bring the sister-chromatid together with, you need to bring different parts of chromosomes together, right? Do you think this is generally a mechanism here?
I would wish to, but I don't know for sure. Our data show that they're very sticky. So they just like to, if you have enough sumo, no matter where it is, PML body will stick to it. Probably because, Danny showed, PML body really moves very fast. So they get a lot of opportunity to be sticky. So I think it's very likely. But I don't know how that can be tested.
But then you need some kind of directed, transport. I mean, these clusters, they have to find each other, right?
That's kind of what's puzzling me. I'm talking with Krishna about it. Like, how is that even possible? Like gradient, or actin cable, or just I don't know, but very interested in following that. But that's, again, requires this kind of measurement, quantitative measurement that be guided by theory, because we can measure lots of stuff that will not be quantitative enough to actually conclude. So, yeah.
Awesome talk. Question. The Tera system, the Tera measurements, you said knockdown reduces certain proteins.
Yes.
Does it lead to loss of DNA damage repair? Like you have a...
Yes,
Okay.
it leads to loss of everything.
Okay.
So you don't have clustering, you don't have APB, you don't have DNA synthesis.
Cool.
It's very, very upstream terror. Okay. It's very upstream. And we've done now to inhibit knockdown terror and inhibit sumo, and the knockdown terror even has stronger effects. So we believe terror actually even upstream of sumo. And
wow.
then by default, it's upstream of PML body formation. So you definitely need terror, but we don't know if it's because of the transcription itself or the RNA itself. That's why we're looking for RNA binding protein to show that it's the RNA binding proteins that are important.
Okay.
So it could be transcription too.
So maybe this is a stupid question, but should that be called a different condensate, or is it still PML body once it merges? And, you know, maybe it actually has a different composition?
I asked Simon about his insights on how do you call those different DNA repair condensate? Are they sequentially just different one condensate changing composition over time is just different condensates. One dissolved, the other one come do the job. So I don't know.
Yeah, but here they merge, right? The two condensates merge.
Oh,
so
when
they
the two condensates
yes.
merge, you mean? Oh, sorry.
Yeah. That should be called a new condensate.
Yeah. So, well, they're called APBs, but they're actually PML bodies. So, but when they merge, yes, because the telomere itself is different from the PML body.
More questions? I still wondered, you said there's multiple telomeres in these condensates.
Yes.
Is it at one point a problem of space that if you put too many of these chromosomes around each other, sort of that they interfere?
That's a very interesting question. So we're trying to do this artificial clustering over a very, very long time. We don't see a plateau. It just keeps going. We don't know how. And then the rate is not even exponential, just kind of the same rate keeps going. I don't know, like, looks like there's no limit, but for the cell, there's a limit because they have to finish everything in G2, then go to mitosis, otherwise they're stuck, right? So for the cell, the time is about like 4 hours or something, and but artificially, we can allow them to cluster 10 hours, the limit of imaging, and they still cluster, which is bizarre. So, yeah.
Thanks. Are there any more questions? Otherwise, we move on to David.